black hole. The DNA exerts gravitational force. I've talked about this for 25 years, more or less, but I never really explained why it does that. The DNA has the dodecahedron pattern in it. That's a resonance pattern. Just like any sand that you had in that drop of water is going to get attracted into the geometric pattern. If you drop more sand in from the top, it's all going to get stuck into whatever geometric pattern it's making. So that DNA by itself, its simple existence there, is attracting in all light. And the really strange part, as I said, is the third tube, because the third tube is what happens when you take the DNA away. The light continues to stay in the position of the DNA for not 1 day, not 5 days. I'm starting to feel like I'm an auctioneer now. Not 10 days, not 15 days, not 25 days, 30 days. There's no DNA in the room, but the light is held in place as if it was still there. That's got to be one of the weirdest discoveries in all of science. And it doesn't really make sense until you see that the DNA is one of these geometric forms. It's a geometric resonance pattern, and by simply staying in that space for a while, it conditions the space. You can remove the molecule, and it's trying to make new DNA. That wave pattern is still there, and it's attracting whatever is around it, in this case just light, because they don't give it anything else. But I would wager that if you actually put other stuff in the room, that it would also get attracted to the pattern of the DNA. And this poses the possibility of very interesting technological breakthroughs that we could make, in which, if you really were able to think about this, your body is all one big piece of DNA, right? It's just all trillions and trillions of DNA molecules. I'm standing here right now, but later I won't be here. But the theoretical implications of what I just told you is that a ghost-like image of my body will be here for 30 days, right through whatever other shoots happen in this room. And if you had the right equipment, if you could get it really, really dark in here, and you could count those photons, you would see the image of my body wherever I stand the most. There's going to be a ghost of me. It's going to be very, very faint and very hard to detect unless you had really good optics. But you could do it. You could have a whole room full of people sitting for a lecture, and they all leave, and if you had the right equipment, you could photograph where they were for up to 30 days. That's the awesome implications of this discovery. So if DNA is doing this, if it's absorbing light and holding it, then light is obviously very important for the DNA. This gets into the work of Dr. Fritz Albert Popp, as I've said, who discovered that DNA holds about 1,000 photons inside of it, like a fiber optic cable. And what he discovered was that cancer cells are cells that don't have light in them anymore. A carcinogen, a cancer-causing chemical, is a chemical that blocks ultraviolet light from getting into your DNA. So now we have a totally new model of health and healing, photonic biology, where the photons determine how healthy you are. And guess what type of food has the most amount of photons in it? Vegetables. You have 10,000 photons per square centimeter on vegetable matter as opposed to only 1,000 for animal tissue. So vegetables are loaded with this light. And since it's dark inside your body, you actually have to eat vegetables because you got to feed your light body. There is some part of you that consists of all that light in your DNA, like a ghost, and that might be some other part of you that exists away from your physical body, as this idea of the soul. Maybe the pattern of light is the soul, and those photons are held by your DNA, and as they go in there, they're cultured by that resonance. There's a geometric resonance there. So all of this, again, is very, very fascinating, but where it really comes into play is the idea of ascension. The idea that maybe DNA isn't so solid and fixed. DNA could move around. And all you have to do is remember what I said about the fullerene, right? The fullerene, you slam it, and it flips over somehow into this wave, and it doesn't have any solid mass at all. So maybe the DNA is always flipping over, and as it flips over into a wave pattern, pieces of it can rearrange and go from one place to another, so that when it reconstitutes, it could have a different pattern, a different structure. If you introduce a different frequency into the DNA, then it might go into a wave phase, and then when it comes back to a particle phase, now all of a sudden, you have a different organism. You might be able to intelligently beam the right patterns in, and now you have a model of why our solar system might transform as we go through this epic solar flash. That's really very interesting to me. So let's review this. We're looking at the DNA molecule. We see a totem pole of dodecahedrons, gray and white. And we notice that the double helix is wrapping along the edges. And again, I like to think of this as another good analogy would be like a stream. When you say to yourself, let's look at the rocks at the bottom of the stream. How did the rocks get there? Did the rocks attract the water? Were the rocks already there and then the water is magnetically attracted to rocks? No. What happens is that it rains and the rains are pouring down the mountainside. The rocks come out of the dirt, they get caught up in a flow of water, and they roll down to the lowest point. They roll into the bottom of the stream, which is where all the water is draining. So the rocks didn't start there, the rocks got pushed there. So what I'm saying is that DNA behaves the same way. That DNA is caused by a life-forming wave, a life-giving intelligent energy that is all over the universe, just like UCLA is now saying, and it's pushing atoms into that position. And now we're back to Dr. Luc Montagnier, a Nobel Prize-winning biologist, discovered the HIV virus, and maybe to work off his karma, he came up with this amazing discovery where you have DNA in a sealed test tube and a regular test tube of ordinary water next to it. You zap them for 18 hours with 7 hertz frequency, which is the basic electromagnetic energy that the Earth gives off. And the water in the tube that's just plain water has DNA grow into it. And if you're like me and you heard me say this before, you're scratching your head going, well, wait a minute, David. Where the heck did that DNA come from? Did it melt through the wall? No. What I'm saying is the water molecules got rearranged on the quantum level to become DNA. They had transmutation like alchemy. And that totally gets back to this idea of the buckyball slamming against the wall, turning into a wave pattern, and then reconstituting. Those atoms of water are turning into waves, and when they get new information, those little particles in the waveform are pushed back into a new form. So when they crystallize, now they start to form DNA. And this is probably happening trillions of times a second, and it coalesces DNA where DNA wasn't going to be. Montagnier's experiment is another one that you have to bring up every time we talk about this because it so elegantly shows life is going to show up everywhere. All I'm doing is taking really highly credible scientists, Nobel Prize winners, and PhDs and MDs and putting their work together and having it all make sense. And even when we photograph the DNA molecule with an X-ray diffraction image and getting a little microscopic picture of it, we still actually see very compelling geometry. This is, you can look it up on Google, it's pretty easy to find. Just look up DNA diffraction, X-ray diffraction. This is what DNA looks like when you actually zap X-rays through it and you take a little quantum photograph of it. That looks way too geometric compared to how DNA is typically shown to us, but it all fits together within this idea that DNA is a resonance pattern, that DNA is a crystal of this resonance that's in the universe. Now, I don't want to stop on DNA because DNA only takes us part of the way there. Dr. Mark White, who is a medical doctor, has actually cracked the code of life and found a geometric underpinning. Now, what do I mean by that? Before we get any deeper into this. So let's break this down a little bit before I get into the specifics because this is important. Somebody could argue, well, David, okay, so you got Montagnier who shows that DNA shows up in water. But if you prove that DNA can form that way, you don't have a full biological model, dude. You've only got DNA, which is just like a crystal anyway. That's not going to form life. Okay, well, let's talk about that. What do you need to form living tissue? Living tissue is made of protein. All the different types of tissue you could ever have are proteins, and we know this. Basic biology. What are proteins made of? They're made of amino acids. What is DNA made of? 4 basic types of amino acids. So now what we have to do to take life and make life exist, to make living stuff happen, You have to build proteins, which means you have to build amino acids, and then you have to combine them in the right ways so that they will actually form protein molecules. Now, this is where it gets really interesting because the relationships between amino acids are very, very complex. It's like a whole family reunion in which there's all these animosities, and certain people are going to want to talk to each other, but other people will turn their backs on each other and never want to talk. Then you have, in the case of the amino acids, you have some that really like to be in water, and you have some that really don't like to be in water. They all have these complex interactions, and it was literally discovered in the late 1960s by brute force experimentation how certain amino acids either make handshakes or don't, and they're all different. And there's 20 core amino acids that are really the key to how this all works, as we're going to explore. So those 20 amino acids, they all have different relationships. Some of them will bond, others will repel, and it gets very complex. So when you typically see a biological chart describing this, as I'm going to show you in a minute, they just lay it out in a nice square, boxy diagram. And they have the idea that these relationships exist, but they don't know why. They don't know what's really going on. They don't know why certain things like to talk and other things don't. It's all just something that they did by hunt and peck. Let's put these two together, see what they do. Okay, this seems to always happen. Put these two together. That always happens. They don't know. Dr. Mark White is smashing this idea that it's simple, because it's not simple, it's complex, but it's complex in a geometric way. There is a geometric, unified field theory of biology that solves all the mysteries, and that's what's so amazing. So first I want to show you this picture, and this is Dr. Mark White all the way over on the left. He's holding a piece of paper that has a diagram on it that you can't really see too well, but that is a diagram of the dodecahedron pattern that he found in the amino acid relationships. That's the key. And he's there with some other scientists, and notice they're all holding little geometric objects. Well, Dr. Mark White independently discovered the same thing we were talking about with DNA. This diagram is a lot harder to see than the one I was showing you before, but he mapped out the relationships of DNA, the double helix, and he stacked these dodecs on top of each other, and sure enough, it all lines up beautifully. So that's his own geometric visualization of how DNA is formed from this geometric resonance pattern. Now, his website is called codefun.com. I am not intrinsically a biologist. What I've done is to find things that I can understand up to a point, but others are more specialized in these fields and will be able to take it to the next level. Mark White is very accurate on his website in saying that this is a scientific revolution in the making. This is as important as Newton or Copernicus or Galileo or Einstein. I do believe that. And I don't have the expertise to fully understand all the nuances of what he came up with, because it is very, very complicated. And he has a lot of papers on codefun.com that you can take a look at. But there is one page that you get to when this all starts, when the discussion originates, and I want to read you a little bit off of that page now and show you the page where he talks about how this works. So it says, the genetic code dogma has utterly failed. And again, that's just because the amino acids, they don't really know why they fit together the way they do. They've just mapped it out, but they don't have a model to explain it. And he says, introducing the perfect code theory. I'm going to read you the rest of the stuff in the middle there, most of it, but just look at the bottom. He says, what is the ideal form of a DNA molecule? It is a double helix. What is the ideal form of the double helix? It is a dodecahedron. Bang, there it is. He's got it. What is the ideal form of the genetic code? It is also a dodecahedron. Let's hold it right there. So the ideal form of the genetic code is also a dodecahedron. That's heavy, because again, this guy's an MD, and we're going to see that Russian scientists independently rediscovered the same thing, which is really amazing. And they found his work thanks to the power of the global brain of the internet. The Russian scientist's daughter actually sent him these papers showing that this guy actually discovered the same thing. So if the ideal form of the double helix is a dodecahedron, and the ideal form of the genetic code is a dodecahedron, with a little modification, then that's fractal. That shows that the laws that make proteins out of amino acids are a fractally larger dodec than the same dodec that's making DNA. Are you with me? This is heavy stuff. You got this resonance pattern making the double helix down at the quantum level, and you expand it into a larger size, you resonate it, right? As you get resonance, you get ripples, just like you drop a rock into a pond and you get ripples and they become larger and larger rings, but they're all ring-shaped. You get larger and larger dodecahedrons. And the same laws that are down there at the DNA level, they're encoded in those dodecs. Those dodecs have little fractal twists and turns in them. They're not straight dodecahedrons. They have all this little extra energy, and that could be the real secret of how these complex proteins are forming, just like he discovered. Well, that's amazing. And so I'm not going to go too deep into the geek speak here because it's very dense, but I want to show you some of these diagrams of what he came up with. And first I want to read you what he said on that main page. The first step toward properly understanding the genetic code is to realize that nobody actually understands it. And this is very important. The second step is to realize that the genetic code is a dodecahedral molecular language. That's really, really cool. Now, what we're seeing here is somebody who built a dodecahedron based on the same kinds of models that Mark White is talking about. In other words, if you look up at the top of that dodecahedron where it says WA, and you see that one of the corners says the number 20. Well, do you remember when I was talking to you about the fact that the atomic nucleus has these protons that are corners of a geometry? And what did I say the number of corners were on the dodecahedron? 20, right? Because you go from the icosahedron at 26 protons to the dodecahedron at 46, because you get those extra 20. Well, there's 20 key amino acids. That's all there is. So what he did is he takes those 20 amino acids and he sticks them on the 20 corners of a dodecahedron. So what you're seeing here is a cardboard model of somebody duplicating what he did. And sure enough, when you actually put the 20 amino acids in those positions on each of the corners, so like look at the front and notice in the middle there you see the 15 on the top, which is the bluish one, the 15 on the left, which is the reddish one, and the 15 on the right. They're all pointing towards that corner in the middle. So each corner has a number, and those numbers are all going to be the same on each of the three faces. And there's only 20 of them. There's your 20 amino acids. And they want to combine. Now he's got this really interesting model in which that's the kind of core matrix of it, but then how the DNA interfaces with it has to do with this tetrahedron. In other words, you can take a tetrahedron and put it inside a dodecahedron. It'll fit perfectly. And he talks about how there's 120 different ways that you can turn that tetrahedron, which is very beautifully elegant for the making of life itself. Now, believe it or not, he also actually sells toys, and I'm not getting any endorsement from this, but just to show you how far this has gone, these are soccer balls with his other business name, Rafiki, on it. So there's all these cool things you can buy that show the different types, the names of the proteins, and where they appear on there. So you see on the left, there's a blue triangle that says lysine, all these different things. So that's the map of what he's done. You can buy that on a soccer ball. Now, all of this stuff, again, his model builds off of the basic 4 types of messenger RNA. Remember I was talking about how there's only 20 of them, and they normally are in a nice, boring square diagram? Well, here's what it is, and this is taken right off the internet. The genetic information in messenger RNA is composed of an alternating sequence of the 4 bases, adenine or A, guanine or G, cytosine or C, and uracil or U. That's all there is. There's only 4. And it's all about how they combine. And again, nobody really understands why there's only 4 and the rules of how they combine, but Mark White figured it out. Now let's keep reading. So you have A, G, C, and U, and the way they combine provides the unique code specifying each of the 20 basic amino acids naturally found in protein. That's all there is. And then, down there, you could read the rest of that on your own time. We're not going to need to do that. So I want you to look at the bottom where it said using these techniques, which is, the 4 basic proteins and the 20 amino acids and how they combine, the genetic code was solved, and the main contributors were Nierenberg and Khorana, who won the Nobel Prize in Physiology or Medicine for 1968. So Nierenberg and Khorana have one of the top 3 most cited scientific papers in the world, because they took these 4 little guys, A, G, C, and U, figured out that they fit together into these 20 amino acids, and then those, in turn, combine in all these funny ways, the 20 different relationships which again are the 20 corners of the dodecahedron. That's what he was missing. Okay, well, wait a minute. What about A, G, C, and U? Why are there only 4? You want to get this? Take a look at this. This is what Mark White found out. Boom. It's a tetrahedron. You see that? Tetrahedron has 4 corners, A, G, C, and U. And that tetrahedron is moving around inside the dodecahedron. And the different spins that it has, of which there's a total of 120, determine all of the laws of the genetic code which nobody has ever been able to solve before. And you can build each face of these tetrahedron triangles with the basic relationships as he shows here. So these are the faces now. So you have the primary, in which it's all just one thing. You have the secondary, where there's 3 of them crossing over. And then you have the tertiary, where there's 6 crossing together. This is all part of his model. You take those triangles, and that's how you end up with these various tetrahedrons. And here's a diagram where he's showing how the tetrahedron fits inside the dodec. It's a little hard to see, but notice there's a green triangle on the top of the dodec on the left there. It's actually there in all 3 diagrams, and there's little blue ones popping out from the bottom. That's the tetrahedron stuck inside. He's got other diagrams that make this easier to see. Now here's another diagram off of his website where he's showing you a water affinity scale on the left. Hydrophobic means scared of water. Hydrophilic, philia, is love. Hydrophilic means it loves water. So notice then you have numbers. And the numbers correspond to the 20 basic amino acids. And if you want to pause this and take a look, you have all the names there: isoleucine, phenylalanine, valine, leucine, all on down. I'm not going to read them all. Tryptophan, number 6, you know that one. That's what makes you sleepy. These are the basic amino acids that everything is built out of. You can see down there, number 17, glutamine. People take L-glutamine as a supplement. Aspartate, that's aspartame. It's got that funny sugary taste. It's actually really bad for you. So also notice here that he's assigning each one of them a different color in a rainbow kind of spectrum, and those colors are all different. And so that allows him to map them out as a geometric pattern. You got your pK ratio. You notice they all have a different pK ratio. Then you have their position 1, position 2, position 3, showing how those actual amino acids combine together. And there's only going to be 4 of them, A, C, G, and U. So this is all very cool because, again, the real key is that you got this tetrahedron inside a resonance pattern, and it rotates through these different shapes, making different types of tetrahedral relationships to the dodec that is surrounding it. Now, does this literally mean that life has these little geometric shapes inside of the cells making proteins? Not exactly. What we're seeing here is the underlying resonance pattern that's forming proteins. And remember, that resonance pattern could exist without any matter there, and just like we saw in Montagnier's experiment, it could take ordinary water and start to assemble proteins, which we saw. If you go back to the work of Wilhelm Reich, he was zapping things like pure rock in water with electrical current and getting life to form. And we saw how that was being replicated, and it really works. You can get complex, multicellular little organisms growing out of nothing but sterilized water. And that was our little test tube experiment that was redone, where you see these little guys growing in the water. And that goes way back in earlier episodes that we were doing. I never really gave you the geometric unified model of how that works and how it's all the same laws that make matter, the same laws that create vibratory patterns in a drop of water with sand in it. This is heady stuff. I mean, obviously, you might want to watch this episode a few times to really let it sink in, because it's trippy. But Mark White has some beautiful diagrams. So take a look at this one now. This is his map of how, remember we showed you those different types of triangles, right? You had the one that was red, and you had the one that had like the strip through the middle, and then you had the one that had the 6 triangles. Well, there it is. You can see it unwrapped on the right, those 3 different types of triangles, and then how it wraps together in the actual, it's either an icosahedron or a dodec. It could be either one, because notice you can see the icosahedron there as well from the triangles. The important point is, this is the big secret to the genetic code. And here's another weird diagram from his website showing how all of these different colors and the various amino acids, again, there's only 4 of them, A, G, C, and U, how they all fit together to make these different shapes. Here's another thing we're not going to get into. This is his rainbow spectrum of how the different amino acids all fit together. Here's another thing we're not really going to get into. Notice on the left there, he's showing you the tetrahedron, and he's showing you the hydrophilic side, which is, again, loving water, and the hydrophobic side, which hates water. So depending on where you are in the tetrahedron, it's going to be more soluble or less soluble. And then again, he's showing how it resonates and spins inside the dodeca. Here's another really cool one where he's talking about those little triangles on each face of the dodecahedron and how the actual movements that they make, the forward shift, backward shift, flipping inside out, inversion, complementing, and mutation, all of that stuff is perfectly solved in his model. So here is a view now where he's unwrapped the dodecahedron on the bottom left. You're going to see that a little more in just a second. And then how it fits together on the right. And he's showing how this geometric law actually starts to form amino acids there. You see the triangle. This says amino acid, transitional RNA, all that stuff. This is very, very deep stuff. And here is his paper, the G-ball. That's what he calls this thing that he discovered. Copyright 2007. And in that paper, he shows you the DNA molecule again. And this is the big one. This is the unwrapped map of the 20 different amino acids, as we just showed you. There's only 20 of them. And you got your A, C, G, and U. Those are at the centers of each of the pentagons. You see that? That's where the big letters are. And there's only going to be 4 of them. Then he's got little 3-letter shorthand names for all of the 20 amino acids, and their relationships perfectly map out this way. Last but not least, this is a diagram off of his site from that paper that I just showed you that's really exciting. This is where he really lays it down, some of the simplest wording, and I just photographed it right off of there so you can read it. He said, there is a suitable relationship between a dodecahedron and a tetrahedron, and it is literally identical to the relationship between nucleotides and amino acids. And again, the nucleotides are those first 4 letters. What are the odds? Obviously, it's not. It's not a coincidence. This is what it is supposed to be. This is how it works. Now check out the next one. And again, there's some geek speak here, so don't get too confused. But he says, for all of the conformational isomers of a tetrahedron, meaning the different ways it moves, a single dodeca can contain exactly 120 unique tetrahedrons, which he then maps out there, showing you how that's all contained inside the dodeca. And then, by using simply a system of 4 colors, a sequence of any 3 colors can be used to specify any one of those 120 different tetrahedrons. So an entire language exists between these 2 basic structures, the dodeca and the tetrahedron, even before a single molecule ever randomly tumbles by to take advantage of it. That is really amazing stuff here. Because what we're seeing is, this is the geometric resonance pattern that makes you, that makes me, that makes all of life. It is totally amazing. It is so out of the box. It's so exciting for me. And I have never really taken the time to do this properly. That's one of the great things about wisdom teachings, is I have to come up with new shows every week in perpetuity, basically, 52 episodes a year, unless we take a little break, which we finally did recently. So it forces me to go back and explore some of these things more. And now I feel this is the definitive airing that I personally have done, showing you how this works. In the next episode, I'm going to show you how a Russian scientist independently discovered exactly the same thing, and I'm going to show you the amazing work of Dr. Glenn Ryan from Institute of Heart Math, who is one of the pioneering scientists in the world in this whole area, and his theory that DNA in between nucleoles inside the nucleus of the cell, that DNA is curling into a perfect sphere with a hole in the middle. That's the toroidal DNA model. And that's where it really gets cool. So much more coming up here on Wisdom Teachings, bringing you the information you need that will set you free. Thanks for watching.